Oui, on peut plus. Jean-Louis, non plus. Il souffle. Ah mince.
5 sur.
Après, on a toujours la peur. Parce que bon, déjà, j'avais.
Désolée hein, pour ce contretemps.
C'est. Je vais vérifier que ça soit transféré.
Ah, d'accord.
Ok, c'est bon. Ok. Ça s'appelle Toulouse.
Toulouse.
Un prix commun. Non, c'est parce qu'il demande comment on le met là-bas.
Alors ça, moi, j'ai jamais su.
Je sais pas.
Vraiment, j'ai jamais su.
Franchement, c'est
Alors, lecteur. Toulouse 24. Clac. Je le mets sur mon ordi, mais après, je l'effacerai. Hein.
Comme tu veux. C'est possible de le mettre sur prise interview.
Normalement, ouais.
Ça, je sais ce que
la suite.
Diapo suivante.
Ouais. Affichage, il me faut le mode présentateur. Non, non. Non, mais tu sais pour qu'il est le. Ah, mais c'est bon.
Ah, impeccable.
C'est bon. Attends, hein. Parce que j'ai pas partagé encore.
Je peux enlever ça?
Ouais. J'ai pas partagé encore. Mettre en grand. Partager. 2. Partager. Est-ce qu'il est entouré de... Je sais pas ce qu'il fait. Je vais y arriver.
Yeah, looks good.
Ça a l'air bien. Ceux qui sont en visio, je ne sais pas s'ils entendent quelque chose. Ceux qui sont en visio, est-ce que vous entendez?
Oui,
Est-ce que vous voyez
coucou
bien le diapo?
Cécile. Ouais, coucou Cécile et Isa, on entend très bien.
Le sound?
Je vais vérifier. Ah, c'est bon.
Par contre, Cécile, on voit pas du tout la salle. Hein. Je sais pas si c'est normal, mais.
Isabelle, je vois que tu as ton micro ouvert, mais on ne t'entend pas.
Ah, mince.
Je suis pas sûre. Hein. Je ne sais pas s'ils me voient, s'ils m'entendent. Ouais. Vous n'entendez rien? Augmentez le volume. C'est bien comme j'ai bien le volume au max. Et quand ils te disent zoom, d'ailleurs, il me demande une connexion là. Ça, c'est pas grave.
Cécile.
Non, ça, j'en ai pas besoin parce que j'ai pris le lien direct qui était sur le site. Je sais pas. Ce qui m'étonne, c'est que je le vois pas en tour de rouge. Ah oui, tu l'as pas en présentation. Oui. Oui, tu l'as pas. Ou si tu as escape. D'accord. Si tu vas au partage d'écran.
Ouais, c'est ce que j'ai fait.
Après,
Ouais, je voulais juste regarder
ok.
si, je voulais juste regarder s'il n'y avait pas d'autres. Ouais, ouais. Je voulais pas déjà prendre comme ça, tu sais, je voulais là.
I can see that it's the same everywhere.
Yeah, it's international.
Happens all the time.
Là, je pense que
Là, je pense que c'est bon.
il faudrait juste que quelqu'un me fasse un signe dans le chat. Tu peux appuyer sur le chat pour voir ce qui se dit?
Allez, c'est un passage. Oui, 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 tout est OK. Ah,
Ben voilà.
vas-y, alors.
C'est bon?
Ça va être bon. J'essaie juste de vous repasser en...
Can you move this? Yeah. How do you get rid of this? No.
Mais non. Ah, on va y arriver. Où est-ce qu'elle est passée?
T'as perdu quoi?
En fait, on a appuyé sur un bouton, il ne fallait pas.
C'est bizarre qu'elle n'est pas la vidéo. C'est parce qu'elle est là?
Je sais pas. Il disait tout est OK. Clac, j'ai retourné.
Ouais, parfait.
On est bon?
Excellent.
Allez, super.
OK.
Bonjour à tous. Hi everyone. It's not every day that one has the chance to or get the chance to introduce someone that really shaped modern genetics and development biology. So Professor Norbert Perrimon has turned fruit fly, so Melanogaster, Drosophila melanogaster, into one of biology's greatest heroes, huh? Because from Del 4 US, I can't say this in English, he knows it. That allows precise gene expression to pioneering RNA interference techniques. These contributions have become essential tools that many of us rely on today. So he's a professor, as you probably know it, at Harvard Medical School. And also he explained us that he's also an investigator of the Howard Hughes Institute, and you can combine both, which was interesting for us. So as you can see, and I won't go into the details because his CV is larger than the day we meet. So I won't go more. I think this is a chance for us to have him. We had a really cool discussion this morning, and I hope you'll have a great presentation, and I'm sure about that. So please join me in welcoming one of really the true pioneers of modern genetics and a scientist really whose impact continues to inspire, Professor Norbert Perrimon. Merci beaucoup d'être là. C'est vraiment cool pour nous.
Thank you. Thank you for the invitation. Great. I'm glad the technical things are okay now. So yeah, so really enjoyed my visit here for a couple of days and learning about the place and the science being done here. And thank you for such a great post. So what I want to do today is to tell you about some of the work that we're doing in communication between organs. And for the past 10 years or so, my lab is really focused on to develop models where we can really understand the way the tissues of the organs really interact. And so we're not really specifically interested in one organ in particular. I mean, we really want to take a very system-wide approach to understand the way the muscles, brain, fat, and those tissues, gut, and so on, the way those different tissues are talking to each other. And so most of the studies we do are in the adult, in flies, as you heard. And what our goal here is really to identify the new hormonal system, because we already know about some hormones, like insulin, and glucagon, and so on. But we believe that there are many more adipokines, myokines that remain to be identified. And we believe that we want to use the fly system, the genetics, the powerful system that we have to manipulate gene functions, to really identify the new hormonal systems and really understand what those different hormones are sensing, whether or not it's an acute signal or a chronic signal, and whether or not they can sense, for example, different diets, or whether or not they can respond to different type of environmental stresses. And because we also study this in the adult, a lot of it is about mobilization of nutrients. So we want to understand how those different hormonal systems are going to coordinate the use and the storage of nutrients. And yes, and we want to do this in normal physiology of the fly, but also in different models, models of diet, obesity, aging, and as I'm going to show some model of tumor, where the tumors is affecting the entire animal system. So, so this is a very simplified view about the way organs are, the way they are specialized, and this is really just to give you a sense again about the main communication. So the food is going to get into the stomach and intestine, and the nutrients are going to circulate in the blood. There is going to be storage of nutrients in the liver, adipose tissues, and then the peripheral tissues, depending on their requirement, what they need in terms of energy, are going to send signals to the reservoirs of nutrients for their release and so on. So the point here I want to make on this very general slide is that again the organs are very specialized and really are part of the global network of and then as a result there is going to be a lot of exchange of different secreted proteins as well as metabolites, which are going to coordinate this that system. So so this is a slide here I took from a review we wrote a few years ago, trying to really synthesize what is known about different factors which are being secreted from different organs. So this is really a view from the mammalian system, and you see that it's already complicated. I mean, there are a lot of different hormones which have been identified, like those GDFs, interleukins, FGFs, and so on, TNF alpha, and there are arrows in all directions. And so the system is quite complicated, but I think this is only part of the story. There are many more hormones, we believe, that remain to be identified. And importantly, in many cases, it's not really clear what those factors really act. There's actually a lot of papers which have been retracted about some of the function, what was thought about the way some of those factors are really what they do, where they come from, and so on. So it's a bit of a mess, and we believe that we might be able to use the fly system as a way to sort of help understand the complexity of factors which are being produced by different tissues and are being regulated. So this slide here is simply to make the point that the fly has organs which are very similar to human organs. I mean, flies have muscle, they have adipose tissues, which are called the fat body. They have a kidney, which is called the Malpighian tubules. They also have the liver cell, which are called the inocytes. And they also have a very specialized cell, which are producing like insulin or glucagon. Those are called the insulin-producing cells, which are a set of cells in the brain. And the glucagon are produced from a set of cells called the AKH-producing cells, because the glucagon in flies is called AKH. And those are from those corpora cardia cells, which are located in a small gland. So the fly organs are conserved. And so, yeah, importantly, I mentioned that flies do have insulin and glucagon, which are really central hormones to regulate anabolism and catabolism. And as I mentioned, the insulin is producing from those IPC cells, which are the equivalent of the beta cells in the pancreas. And the AKH glucagon is made from those corpora cardiaca cells, which are also known as, which are reminiscent to the alpha cells in the pancreas. And insulin and glucagon either promote nutrient storage or promote nutrient mobilization. So those are really very central hormones. But in addition, there are many other hormones that you find in the fly, which are also reminiscent to to the human hormones. So for example, a few years ago, we identified this UPD2 hormone, which we showed is the equivalent of leptin, and there are additional hormones like the activins and so on. Okay. So the organs are conserved, many hormones are conserved. And one of the advantages of working with flies is that we can really do genetic screens at scale. So for example, here what we can do, if we want to identify secreted factors, let's say from the brain, that are going to affect let's say the adipose tissues, we can really target a specific knockdown or knockout of the genes of interest in one tissue and look at their effect in the peripheral tissues. So the way we do this is to use this GAT4-UAS system where we're going to express GAT4 only in one tissue, and we introduce as well an RNAi reagent under control of UAS sequences. So this really allows us to downregulate the activity of the specific genes in the brain. And then, depending if we have a readout in the peripheral tissues, then we can identify all the RNAi perturbations which are going to affect the activity or whatever the reporter is in the peripheral tissues. And we can really do this at scale because we have RNAi reagents against all the fly genes. So we have so it's very easy to simply identify, let's say, 500 secreted proteins which are expressed in the brain, and find the RNAi lines that correspond to those genes, and then do a screen to identify their effect on the peripheral tissues. So we have many reagents and tools for doing this type of analysis, and we can do loss of function screens, but we can also do gain of function screens, and using additional type of reagents that are available in the fly. So using this type of approach over the years, our lab has made a number of different contributions, identifying a number of different hormones which are produced from, let's say, muscles to the adipose tissues. And here we have, for example, we have some stories on some of the activin beta, myoglobin, and so on, as well as some PDGF, VEGF, cytokines which are produced from muscles which affect the lipid synthesis or the lipid breakdown in the adipose tissues. And so our work, as well as a lot of work from other labs, is trying to really providing us a map of the different hormonal systems in the flies between the different tissues or organs. So today, what I want to do is to focus on one system, because again, I don't want to spend time on this on some of those studies, but I'm going to be focusing on one model that we have in the lab that we really like, because it's really first of all, we think it's relevant to. We think it's a model for cancer cachexia, which is cachexia is a syndrome of when people have cancer, at some point they're going to start losing a lot of weight, and in most of the different organs. And we can mimic this in the fly. The way we do this is that we generate, in this case, tumors in the fly gut. The way we do this is that in the gut you have a number of different stem cells. So what we do is that we express an oncogene just in the stem cells in the gut. And here those gut stem cells are labeled in green. And you can see that there is so if I showed you the wild-type gut, there would only be a few cells which are labeled in green. But here you can see the very large tumor that has been induced here by this by expressing this oncogene. So it's an oncogene called YAP or Yorkie in the stem cells. So we were able to target, to generate very precisely a tumor in the gut. And so now in those flies, not only there is a large tumor in the gut, but those flies also develop all kinds of organ wasting phenotypes. So if you look in the muscle is going to degrade. In females, the ovary is going to atrophy. The fat tissues are going to be falling apart. And those flies actually develop a lot of metabolic abnormalities, hyperglycemic. There is depletion of triglycerides and glycogen. And in addition, actually, they have this very specific, very characteristic bloated phenotype compared to the wild-type flies. And that's because the flies are retaining a lot of water because their kidneys actually are not functioning properly. So they have this bloated phenotype that we use as a phenotype of this overall organ wasting phenotype. So, so we characterize a number of different factors produced from those gut tumors that affect the physiology and or the integrity of the peripheral organs. And the first factor that we identified was a protein called, a gene called IMPL2. And so this is actually, it shows you the expression of this IMPL2 in the flies which have the gut tumors compared to the control. So there's this huge overexpression of IMPL2. And what IMPL2 is, it's basically an insulin binding protein, which is reminiscent to the IGF-PPs in mammals. And so what IMPL2 does is actually it binds to circulating insulin, which are called the DILPs in flies. And what this does is that it basically it inhibits the DILP. So now they are not able to bind to the insulin receptor and activate signal transduction. So it's really like the way to think about it is that IMPL2 is going to shut down systemic insulin signaling. And so yeah, this is actually to make the parallel between this IMPL2 and the IGF binding proteins. In insects, we have DILPs, which are the insulin-like peptide, which really are the equivalent of insulin as well as IGF in mammals. And this IMPL2 would be the equivalent of those IGF binding proteins. And we showed that the production of this IMPL2 from the tumor is important for those wasting phenotype, because now if we generate tumors where we have removed by using RNAi, for example, IMPL2 from the tumor, now we can partially suppress the wasting phenotype. So for example, here, this is the size of the ovaries in flies which have the gut tumors. So they are reduced extremely compared to wild type. And now what we've done here is that we've removed this IMPL2 from the gut tumor, and we can recover the ovary. The size of the ovary is back to almost normal. And then, and then we also have a number of phenotypes like climbing or as well as the metabolic phenotypes can also be suppressed by IMPL2. So we think that the tumor is producing this IMPL2 to shut down systemic insulin signaling in the animal. Yeah, so this is a model that we had at the time. So it's actually those are pretty old studies already. We think that the tumor produces overexpressed IMPL2, and this shuts down insulin signaling in the muscle, fat, ovaries, and so on. Because insulin signaling is very important for the different organs to have their normal physiology, and that's going to contribute to their breakdown. And one of the reasons we believe that the tumor is doing this in part is simply because as a result, there is going to be less usage of glucose from the peripheral tissues. So in flies, actually, the glucose is stored as trehalose, because the flies need very high level of sugar. And if they add circulating glucose, it would be detrimental to the fly because it's very reactive. So in fact, so the glucose is being stored as trehalose, which are 2 molecules of glucose. And those it's like an inert sugar. So but it's basically the equivalent of glucose, okay? And but we believe that this shutdown of glucose usage from the peripheral tissues allows the more glucose to actually get into the tumors. And what we showed at the time that there is actually an increase in the insulin signaling pathway. So like the receptors, for example, is upregulated and glycolysis is being regulated. So the tumor has adapted as well to this IMPL2 high level in the monarch. Okay, so the first factor we have shown you here is this insulin binding protein, which is derived from tumors. But because we suspected that the other additional factors, the suppression of the phenotype that we see is only partial, we started to look for additional factors. And here we identified another component, and this came out of a chemical screen, actually, where in the lab we were doing a small molecule screen to look for suppressors of either the tumor phenotype or this organ wasting or bloated phenotype. And one interesting compound we identified was trametinib, which is a MEK inhibitor. So at this concentration of 10 millimolar trametinib, you can see that the tumor is not affected, but the wasting phenotype is being suppressed. So this told us that MEK suppression, right, somewhere in the fly was able to prevent the wasting phenotype. So so what the hypothesis then was that that there was some factor derived from the tumor that activated MEK signaling in the peripheral tissues. So when we block trametinib, MEK activity using trametinib, we can suppress this ERK activation. So the first experiment we did following the identification of trametinib was to actually demonstrate that too much ERK activity is actually bad to the muscle on the adipocyte. So what we did is go back to wild-type flies, and here we activated MAP kinase or MEK activity in the muscles on the adipocyte. And this really led to a breakdown of the muscle as well as an increase in lipolysis. So again, this told us that there was some factors coming from the tumor that activate ERK signaling in the peripheral tissues. So in flies, we know actually quite a bit about ERK signaling because years and years of study. So we know all the pathways pretty much that activate ERK signaling. And receptor tyrosine kinase pathways in particular are known to activate ERK. And so because we know all the ligands and receptor pairs that activate ERK signaling, we could go back to some RNA-seq data that we had from the ERK tumor to identify secreted proteins that are known to activate ERK signaling that would be upregulated from the ERK gut tumors. So very quickly, we showed that PDF1, which is a PDGF-VEGF, so it's a cytokine that is known to activate the PDGF receptor and activate ERK signaling. We found that this PDF1 is massively expressed from the gut tumors. So we tested whether or not, in fact, PDF1 could be the ligand that we're looking for, right? And here we did this very simple experiment where we generate flies which have the gut tumors. That's what this genotype means here. And again, they also express GFP, which is why they're green. And then we did RNAi, we co-express an RNAi against this PDF1. And what we saw is that we could actually suppress, again, this bloated phenotype, indicating that PDF1 was one of the ligands derived from the tumor that affected the organ wasting. I'm, of course, skipping a lot of additional evidence for this, but this really, we were able to really implicate that PDF1 was important. So in an additional study, then we also were able to identify another pathway, and I'm not going to tell you exactly how we found it, but we were able to identify another cytokine called UPD3, which in this case, it's like an interleukin 6 cytokine. And those are known to activate the JAK-STAT pathway, which regulate, which is another signaling pathway involved in cell-cell communication. And we were able to show that the tumors produce this UPD3, which activates JAK-STAT signaling, and that regulates this IMPL2 protein as well that I've told you about. So, and we're able to show that this UPD3 also contributed to the overall wasting phenotype. So overall, so far what I've told you is that we've, from genetic experiments using and then validation, we've been able to identify 3 different factors. This IMPL2, insulin binding protein that shuts down insulin signaling, that PDF1 that activates MAP kinase and MEK signaling. But we also know that in some other contexts, it's also able to activate gene kinase signaling. And this IL-6 UPD3 that regulates JAK-STAT signaling. So we those experiments actually took quite a few years to for us to perform. So this was it took about a number of different postdoc years to identify and characterize those factors. So in recent years, we've been interested in trying to see if we could take a much more global approach to really study this communication between different organs more at a more global scale. And for this, we became quite interested in using a single cell RNA-seq. In flight, we do a single cell RNA-seq, single nuclear RNA-seq, mostly because there are tissues where you cannot encapsulate the cells. The muscles, for example, are syncytial, so you have many nuclei, and the cells are very large. So we do everything we do is a single nuclear RNA-seq. And a few years ago, we were part of a consortium where we characterized basically all the other different cell types in the adult fly. And as part of this consortium, we sequenced about almost 600,000 cells that led to about the characterization and annotation of more than 250 different cell types and so on. So we have a map of all the cells in the adult fly, which really allows us now to use this knowledge to characterize phenotype of, for example, those flies which have those gut tumors and those wasting phenotype. So what we did here, so this is really the phenotype of flies as they get older after we induce the tumor. So initially there is no tumor, right? This is the stem cells here would be in green, but there are very few of them, and they are not really visible here, but then they start to grow and grow and grow. And then the phenotypes of the flies are getting worse this time. So what we do now, rather routinely, is that we just grind up the entire fly. So we don't do any dissections or whatever. We just take the entire flies, and then we encapsulate all the nuclei, and then we do the single nuclear RNA-seq at different time points, and we can follow the phenotype. And in fact, those experiments are not cheap, but they're quite doable, because with only about 10,000 to 20,000 nuclei, we can really profile the entire tissues in the fly. So this is a map here of what you get, those whole body map, where you can get a sufficient amount of cells, nuclei per tissue. So for example, here you will have the gut with the stem cells here and the differentiated enterocytes. So we wanted to see whether or not we can use this approach to identify new factors or new interesting candidate genes, which would help us understand the overall phenotype, organ wasting phenotype in those flies. So first of all, we have to make sure that the approach actually works. And here, what we compare here is simply we compare the number of nuclei in the flies which have gut tumors compared to the wild type flies. And here we superimpose the single nuclei from the wild type in blue, the single nuclei from the flies with the gut tumors in red. And you see that the number of cells that really change are those two clusters here, which basically are the stem cells, which are the gut clusters, which are here. So this is a gut cluster with the stem cells and the enterocytes. And you see that there is a lot more red, so which makes sense, because this is where we have induced additional cell proliferation. But then we can go back and we can look at the, So we can go back and look at the expression of the genes I've told you about, right? Which are those IMPL2, PVF1, and UPD3. And what you see first is that in the stem cells, which where we express the oncogene, you see that there is more expression of your key. So that makes sense. And you can see that there is a lot more expression of IMPL2, PVF1, and UPD3 mostly in the enterocytes of the gut. So this is good, because it gives us confidence that what the approach is showing us what we want. So then we started to look for interesting expression patterns, and we tell us something about what's going on in those flies. And here I'm going to tell you a story about two genes, PepsiK and PDK, that we find. So those are two different enzymes which are involved in gluconeogenesis. And we find that they are overexpressed in the adipose tissues, which are the fat bodies. So those are, we are focusing here on the D8 flies, which are those large tumors. And so usually those two enzymes are expressed in the fat body and at a relatively, a pretty good level, but we see that there is a huge increase in the expression of PepsiK and PDK in the fat tissues. So I mentioned that those are enzymes which are involved in gluconeogenesis. So gluconeogenesis is the way you go from, let's say, acetyl-CoA into glucose or trehalose for the fly. And that's the reverse of glycolysis, which goes from glucose to acetic gyruvate and acetyl-CoA. And so what we see is that, again, those two enzymes, PepsiK and PDKs, are like rate-limiting enzymes which are pushing gluconeogenesis. So the more you have, the more you go this way. And you can see here when we look at the amount of trehalose with those D8 flies, there is a very high level of trehalose. Okay? So this told us that maybe I told you that the flies are hyperglycemic, right? And which is due to this high level of trehalose. So we wondered whether or not this actually reflected this upregulation of those two rate-limiting enzymes. So to test whether or not the expression of those enzymes is important for the phenotype, we generated flies. So those are a little bit complex flies because we need to manipulate gene expression independently in the tumor as well as in the fat tissues. So what we do is that we have two different binary systems. One is a GAL4 UEL system, and the other one is a Lexeo system, which really allows us to change the genotype of the flies in the two different tissues. So what we do then is that with one system, we induce a Yorkie in the stem cell, and with the other system, we remove and decrease the expression of the enzymes in the fat. Okay? So that's what this genotype means. Lexea driving Yorkie in the gut, and LQP driving, which is a fat driver, driving PepsiK RNAi in the fat body. And what we see is that in those flies, first of all, we can see that there is a lot less of this bloating phenotype, and there is also a suppression of a reduction in the amount of circulating glucose, the trehalose. So so depletion of PepsiK in Yorkie tumor fat body improves the trehalose level. And so then the question is, what signal is coming from the gut that regulate the expression of those rate-limiting enzymes in the fat? And for this, we've developed a tool which is very similar to a tool that they have that's been developed for mammalian systems, called like cell phone or cell chat. So we developed a version of that called FlyPhone. And basically, what you do is that based on the expression, the genes which are expressed in one tissue versus the other, and using, since we actually know about the structure of the most of the signaling pathways in terms of ligand, receptors, and downstream target genes, we can make predictions about which signal is coming from one tissue to another. So using this, we can figure out, or we can predict or make, hypothesize of that some pathway, for example, one pathway, maybe one ligand from this group of cells or this cluster of cells might signal to another one, and so on. So when we apply this to this communication between gut and fat, one of the pathway that came up was this JAK-STAT pathway, which is activated by these UPDs or IL-6-like protein. So what this the FlyPhone analysis suggested is that one of the UPD ligands derived from the fly gut was going to signal to the fat tissues to regulate the expression of the PepsiK and PDKs. So then what we did is to go back and actually generate flies which had gut tumors, but where we remove PepsiK, where we remove PepsiK. No, sorry, I'm getting things confused here. Yeah. So here what we did is so we use the expression of PepsiK as a readout to test this this cross tissue communication. And we generated flies which have the gut tumors, but we remove now the JAK or the STAT components of the downstream signaling pathways from the adipose tissue. And what you see that when we do this with the JAK kinase, which is called UP here, upscotch, now we don't regulate PepsiK expression and don't regulate PDKs. And we do the same here when we remove, we decrease the STAT, which is a downstream target in the fat. So, so then to really show that the JAK-STAT pathway in the fat is really important to regulate the PepsiK and the wasting phenotype, we now did the similar experiments where we remove now either the JAK or the STAT from the fat, and we measure the sugar level of the trialose. And then we show that, again, there was a decrease in trialose, and also the blotting phenotype was rescued. So what this really told us is that there is from the tumor is producing UPDs, which is like an IL-6, which regulate PepsiKs and PDKs. So those are findings from the flies, but we were part of a large consortium of labs which actually try to study cachexia as a model. And this includes labs who work on mouse as well as humans and so on. And so what we are doing is we're trying to take the findings from the fly and see whether or not they're also they can be validated in other systems. And here I just want to show you a couple of studies that we've done in collaboration with the Genovitz and the Concalves lab, which work either on mouse and humans. And it's a little fuzzy here. But what we do see is that what we're doing here is that there are a number of different mouse models of cachexia, and we are asking whether or not in those mouse models there is, in the cachectic mouse, there is actually an upregulation of the PepsiK or PDKs orthologs in the mice. And we can see that we can detect this upregulation as well here from the cachectic sample compared to the non-cachectic samples. And then in vivo, we've done studies where we now, in those cachectic models, we actually add an inhibitor of PDK, which is called PS10, and whether or not we can actually improve the survival of the cachectic mouse. And you can see that there is an effect here that can be shown in this model. And then similarly in humans, we also find a nice correlation between the level of PDK3 and PepsiK1 in humans which have a cachectic phenotype compared to non-cachectic. Anyway, I just wanted to show this because this is an example of how we can really go from the fly, generate an hypothesis, and then do a follow-up analysis in the mammalian system. I mean, the issue here with all the mammalian studies, and in many cases, it's correlative. There is not. It takes a lot longer to do those type of mechanistic studies as we can do them in the fly, but at least here in this case, those are pretty good leads in terms of components, pathways which are misregulated that contribute to the overall cachexia phenotypes. So overall, this is a summary. What I've done is to pick a couple of examples of studies that we've done on the system, and I've shown you that we have those three factors here. And I focused here on this branch mostly, this hyperglycemic phenotype due to PepsiKs and PDKs. But we have a number of studies on all the different organs. I don't have time to go through this in detail, but we've done very similar type of studies on for example, the fly kidney. And we know that this PVF1 factor is really important to regulate the water resorption in the kidney. We have a story showing that the combination of IMPL2 and UPD3 are really important to regulate fly behavior. So we think actually we believe that this is a very nice model to study anorexia, and we have we've been able to dissect the way some of the anorexic phenotypes are being controlled by those factors. We've been able to show that MPL2 is also affecting some of the stem cell systems and so on. So the idea is really, well, again, what we do in the lab is that we're taking a doing perturbation to different tissues. Again, focusing here on the tumors, but we also do additional type of perturbations. And what we want is to generate phenotypes, different type of phenotypes in peripheral tissues, and then really try to understand the communication of the factors from the damaged tissue, communicate the stress and so on to the peripheral tissues. So so I've shown you about how we use in particular single nuclear RNA-seq to get some insight on what's going on. I should say that we have a number of new secreted proteins that we're characterizing right now that came out of this analysis of those large data sets. But again, I don't have time to focus on this. What I want to do here is to illustrate another method we're using to also try to identify secreted factors, but this is using a proteomic approach. And what we do here is that we've developed a method where we expressed a biotin ligase, and in this case, it's a promiscuous biotin ligase, which is going to be that we are going to express in the ER of one particular tissue. So the idea is that we express, let's say, only this BRA enzyme, biotin ligase, in the fat, and there is endogenous biotin as well in the flies, but we add a little bit, we feed a little bit more biotin to the flies. And what this does is that as a result, all the secreted proteins which are from the ER of the fat cells are going to be biotin related. So now what we can do, we can either isolate the fly blood, or we can even dissect peripheral tissues. And then using streptavidin pull down, we can identify all the biotin-related proteins which are derived from the fat tissue. And so this allows us to identify at scale, again, the secreted factors. So we perform a number of different studies, and here I'm only going to focus on this proximity labeling approach from the Yorkie tumors that I've described before. So again, what we do is to express the BRA ligase in the ER of the tumor cells, and then we identified all the proteins in this case which are present in the blood, which are biotin. So I should say that we do this in wild-type flies, but we also do it from two different tumor models: the Yorkie tumor model that I described, where you have proliferation of the tumors and those cachectic phenotypes, but also from the activated RAS, because here in this case, we see proliferation in the gut, but there is very little cachexia phenotype. But what we want to find are factors which are secreted here compared to here and here. So we're going to be comparing mostly the labeled proteins which are enriched in this experimental sample compared to the others. So when we do this, we're able to find secreted proteins from RAS and from Yorkie, somewhat uncommon. But what was very striking is that almost 50% of the top 100 proteins which are enriched in the Yorkie samples reflect an inflammatory response. And those are coagulation factors as well as innate immunity factors. And what we're able to demonstrate, I don't have time to show you all the evidence for this. We're able to show that most of the factors, first we're able to show that those inflammatory factors regulate the NF-κB pathway, which then regulate the expression of those three different factors that I've told you about, the PDF1, NPF2, and UPD3. And but more interestingly, so we were able to show that so we were trying to understand where are this inflammation. So I mentioned we found a lot of inflammatory factors which are being secreted, like some of the ligands which activate the PGRP or the toll pathways. And we're trying to understand where those are coming from. And what we notice is that there is actually very extensive cell death in the Yorkie tumors. And this is what we detect using this DCP1 staining. And you see that in the RAS tumors which are non-cachectic. There is not that much cell death, but there is very extensive cell death in the activated network. So in green here are the tumor cells, and in red are the dying cells. And what's striking is that the dying cells are the ones which do not express GFP. So which means that so in the tumor, you have some wild type cells and some tumor cells, but the cells which are dying are the wild type cells. And the idea is that they are receiving a signal from the tumor, which are inducing their cell death. And we showed that this actually is that the dying cells are dying because there is an activation of the GNK pathway. And this is a marker called pucker, which is a marker of GNK signaling. And you see that in the Yorkie tumor, there is a lot of expression of pucker, especially in the wild type cells. And in addition, we also see an increase in ROS in the dying cells. Okay. So altogether, and I realize I'm going fast here, but I can provide more information later if you're interested in. What we think is going on is that in the Yorkie tumors, and we believe that Yorkie, we've shown using ChIP-seq that what Yorkie is doing is it's activating this TNF-alpha Eiger ligand, which then signals to the wild type cells and activate the GNK pathway that leads to the death of the wild type cells. And those dying cells are releasing DAMs, like ROS, for example, as well as some inflammatory innate immunity ligands, which then lead to an activation of the so mostly the DAMs are going to lead to an activation of the DNF-kappa-B pathway, which then in turn lead to an upregulation of the cytokines that I've described before, PVF1, that mediate this wasting phenotype. So this is an example here where by focusing on inflammation was pointed out to us through the proteomic studies, we're able to get some insight on the interaction between the cell types between the tumor cells and the wild type cells in the gut that lead to the expression of the different factors. So let's see. How am I doing for time here? I have no idea what. Okay. Yeah. Okay. So what I want to do now is that so my talk is mostly to give you a sense of the type of questions we're asking and giving you a few examples. But we were spending a lot of time trying to develop methods which allows us to really study this communication between organs at scale. And that's where I'm really focusing here on technologies like the single nuclear RNA-seq, the proteomics. But now I'm going to show you some of the, tell you about some of the additional methods we're trying to develop to really understand the complexity of those systems. Because we have one of the problems that we're having is that now we have many new factors, putative ligands, which presumably are involved in these communications. And it's really one of the problems that we have in terms of trying to figure out exactly what all of those factors are. So all those CGs here are examples of genes that we have identified that we know that when we perturb them, let's say in the fat body, we see an effect in the muscle, but those are proteins, secret proteins where we know nothing or very little. And one of the problems is that we don't really know what the receptors or the pathway they're connected to, right? And in fact, here I put this slide here because we made a lot of we were able to characterize a number of factors relatively quickly and demonstrate that their effect was very direct, simply because we already knew what the signaling pathways were. Right? I mean, I've told you about JAK-STAT pathway, activin beta, and so on. But in this case, we already we know what the receptors, what pathways are. So if we're able to show, because to show directionality between two different organs, you need to show that if you remove the ligand here, or if you remove the receptor here, you basically have the same effect on the targeted tissue. But so for the pathway that where we knew the components of, it was relatively easy to figure this out. But for those, we don't really know what their pathways are. So we have, and we have actually a lot of factors. I mean, those are some that came out from genetic screens. But here, again, we've used the proximity labeling to really try to get a list of all the components, which are all the proteins that circulate in the blood. And here, the way we did this is to express this BRA in all the different larval organs and muscle, trachea, fat body, glia, and so on. And then we bled the larvae, got the blood, and performed the proteomics from the blood to identify all the biotin protein. And this really gave us now, we have a list of about 540 proteins, which are in the blood that are derived either from fat, glia, gut, and so on. And many, for most of those, we don't really know about what they are doing, which is why we need to think we are trying to find ways to really identify the receptors and pathways of those secreted proteins at scale. And so I'm going to show you two different approaches that we are working on right now. I mean, the first one is a method using, it's a bioinformatic approach, which is based on the AlphaFold. So you all know about AlphaFold, you predict structure of proteins computationally. And in particular, there is an algorithm out there called the AlphaFold multimer that allows you to predict the interaction between two different proteins. So what we do now is that we take those orphan secreted proteins that we have, and we scan them again. We look for their binders again. So we have we took all the transmembrane extracellular domain of transmembrane proteins, and we ask whether or not there is a predicted affinity between the orphan secreted protein and any of the transmembrane proteins. So we have about, so it's about for one versus about 3,000 or so transmembrane protein. And what this allows us to do is to narrow down the scale, the space, and say, okay, this protein may be binding to those 5 or 6 receptors. And so we were doing this at scale. And I should mention that we have a paper on bioRxiv actually on this for people which are interested. And I'm not going to get into this, but the way you do those predictions, there is a score which is called the IPTM that was described in the initial AlphaFold multimer paper. And what we've done is to develop a slightly different scoring method that we think does better for proteins which have unstructured domains. But anyway, if you're interested, you can look at this. And we're already doing this at scale. I mean, this is an old slide, but now we've done about 500,000 or so different predictions between all kinds of proteins. And so what this allows you to do is to, again, take your orphan ligand and predict a number of likely interactors. And so the first thing we did is to actually see whether or not it works with what we already know. So in this case, we take a DILP, which is the insulin in flies, and we score it against all the transmembrane protein. And you see that, so those are the two different scores, the one, the IPTM, and the LIS, which is the one we developed. And you see that the insulin receptor is the top candidate. Okay, so we're able to really retrieve the non-interactor. Same here for the EGF spits. We can recover the receptor, EGF receptor as well. This protein called STAR, which is a chaperone for the EGF to allow it to get out of the cells. Same here with the FGF, which is called branchless in flies. We can recover the two FGF receptor, breastless and heartless. And TGF beta, DPP, we find type 1 and type 2 receptors, and so on. So we think this approach is really going to help us to narrow down, to make predictions about the receptors of specific orphan ligands. But those are our predictions, so we need to validate them. And for this, of course, we can do conventional biochemistry to predict, to show binding and so on. But I said we have a lot of different factors. So we are trying to develop a method to do this at scale. And the way we're trying to do this is to use a CRISPR activation approach in cell lines. So the idea is that with CRISPR-A, you can activate the expression of any genes that you want. The way it works is that you you target guide RNAs upstream of the transcriptional start site of your gene of interest, and then your guide RNAs, you do this in the context of Cas9, which is a dead nuclease Cas9, but which is hooked up to a bunch of transcriptional activators. So when you do it like this, you're able to bring in upstream of the TSS a bunch of transcriptional activators which are going to give you the turn on the gene which is downstream. So the idea then is to have a library of cells which are expressing all receptors in flies, so each cell expresses usually one receptor. And the idea then is to use the orphan ligand as a bait to pull out the cells which express the receptor that presumably bind to the ligand. And so this is the pipeline we have. We start from a guide RNA library of about 35,000 guides, because we have multiple guides against all the transmembrane that target all the transmembrane, all the genes encoding transmembrane proteins as well as GPCRs. And then we deliver those guide RNAs to a cell lines which have the transcriptional activators. And the way we do this, those guide RNAs, they insert into specific sites that we have engineered in the cells. So at the end, one cell expresses one receptor, and then we pull out the cells using a labeled ligand and then sequence by NGS the guide RNAs which are enriched in the population. Right? So if you do this, you should. This is just showing that it works too. Like in this case here, we use EGF as a ligand, and we're able to enrich for cell population with the overexpressed EGF receptor. So anyway, so this is work in progress here to try to. So again, one of the major bottlenecks we have right now is to identify the receptors of the orphan ligands, and we think that using bioinformatics and this type of high throughput screens, we should be able to identify receptors for many ligands. And I think I have only one more. Just want to show one more method we're trying to develop here, which is to really, basically what we want to do is to develop an ELISA for the fly blood, because we want to become a lot more quantitative in some of the experiments that we do. And we'd like to measure actually the level of many of those different hormones under different conditions, because most of the studies we've done so far is to really take one condition, focus on one tissue, and measure and show that one ligand is really important to mediate an effect. But we don't really look at the way those other ligands are also being affected in the cell perturbation. So remove this factor from the beginning, you never go as bad. Okay, but it's a good question. We can really do use this a trick to make them to recover.
Okay, my question was about the BRA.
Yeah.
You said it has a promiscuous role. What did you mean by that exactly?
Yes, very good. So the wild type BRA, it only biotinylate. So the enzyme, what it does, it takes biotin and it biotinylate lysine residues, right? But the wild type BRA, it does it only in the context of a few amino acids. So only when you have, let's say, I don't know, I'm making this up, glycine, lysine, you could. But the enzyme we use is an enzyme which has been evolved and mutagenized. So that was done in collaboration with and so it becomes promiscuous. It does not really need a consensus. So basically, every time the enzyme see a lysine residue, it's going to add a biotin. Yeah, that's we can pick that up. But it took a long time to evolve those different enzymes. Yeah.
Thank you.
Okay. Yeah.
On the same line, what's the average length of the extracellular molecules that you have identified, and did you do a cutoff to reach 5,000 environments of cigarette free?
Yeah. So usually for the I think what you're talking about is because you can have secreted proteins which are going to be like 20 amino acids, and some which are going to be above. And in fact, most of the annotation is at 100. So all the numbers I show you, it's about at least peptides which are 100 amino acids. And that's really an issue because there may be many smaller peptides, like 20, even 10 amino acids. But those are more difficult to predict because it's, like for example, there are lots of long non-coding RNAs in the file, or anywhere. And you can find a lot of small offs, but those no one knows really if they are real or not. So that's why people have put the cutoff at 100, because if you get an open reading frame of around 100, then you're pretty sure that that protein is valid. And so there is a lot of other type of studies and where people are focusing on the below 100. And in fact, that's actually a problem for the BRA approach, because when we do the proteomics, because there is noise in those different approaches. And so what we do is that we select only the proteins where we find two independent biotin peptides. So if you have very small proteins, it's very unlikely you're going to get two biotin peptides. So we're losing a lot in that space of 0 to 100. So we need to have additional methods for those. Yeah.
Since you're working with gut, is it possible to integrate also microbiota? How comparable it is the fly one to the human one?
Yeah, so the question is about the microbiota. I'm not sure exactly what the question was, but
also integrate microbiota.
Oh yes, yeah. So some of those studies we do it in axonic flies as well. So like for example, I showed you that there was an increase in inflammation and innate immunity and so on. And we know that the severity of the response is greater if you have the bacteria present. So we can see this inflammation in the absence of bacteria, but it's actually less than if you also have the bacteria. So yeah, I mean the bacteria is also good. Microbiota is really going to be playing a role in those different systems as well.
And is it comparable to the human one?
Well, it's a lot simpler. I mean, basically in the fly microbiota, it's the way we raise them in the lab. There is usually like maybe 4 prominent species. It's all about Acetobacter, Lactobacillus, and so on. If you look in humans, we're talking about hundreds of different species. So, but clearly, the bacteria. In fact, we like to think about the microbiota as another organ, right? It's part of this. But it's more complicated to study because it's mostly because the effect of microbiota of the bacteria it's difficult to deconvolute. It's really the mixed population that has the effect. And if you have, it's really difficult to find out exactly what type of bacteria is doing in the system.
Alors, on a perdu toutes les questions qui étaient sur la visio,
On les a
parce qu'en fait, il y a eu une déconnexion tout à l'heure, et donc toutes les questions qu'il y avait sur le chat, elles sont parties.
Uh-oh.
Voilà. Donc, j'ai relancé pour savoir s'il voulait en reposer d'autres.
Okay.
Les care, you can go to your next lesson if you want.
So you said that among the protein that was secreted, there was out that were related to inflammatory system, and all the others, you have no idea of.
Yeah, we haven't. Yeah, it's just those are pretty recent results. Yeah, we have to. And that's really the issue that you do this type of approach. You generate a lot of data, so you have to try to come up with stories that you can finish. So yeah, so I mean, the idea that we'll be publishing a list of things and people can go back and see if they find something interesting. But again, this is why we're spending time on trying to develop methods. We're not doing this to just study one gene. And so, because otherwise we could just keep doing more classical type genetic screening. We would find plenty of single gene to study.
During your introduction, you were talking about physiopathological condition, testing the crosstalk between organs.
Yeah.
So the network you identified with the tumor model, how could it be modulated by aging, for example? Does one factor is taking the first part and the other are down, or the network is totally inverse?
Yeah, I think so some of those factors, I mean, we see them changing with age. I mean, with age you're going to get more inflammation and you're going to have, more flies are going to become more diabetic type. And so you do see changes in like those UPDs or the IUP2s. Those are pretty general factors, and they do play a role in aging. But I think we do have a project in the lab trying to figure out when we do stress, we generate stress conditions, the other different type of stress, whether or not it does mimic some of the changes that you see during aging. So the question is, depending on how we stress an organ in young flies, does it actually start to activate an aging signature? So that's, yeah.
But in an aging context, can you see that a network is inverted? It means that the players are there, but the outcome is totally different.
Oh, I see what you're saying. I don't really have an example of this.
Because of the regulations that is already high, and so do you have some feedback, negative feedback contract sometimes? You can see inverted.
Yeah. Yeah, no, I can see it. But the data sets exist to this time. There is in particular, there was a paper from Ongji Lee a few years ago where they did single nuclear RNA at different time points, 5 days, 15 days, 25 days, and so on. So you have about 6 different time points. So you can potentially take all those data sets and try to do some kind of network analysis and see if this is not a good idea.
For the moment, regarding the secreted molecule that you have, you are collecting them in the blood, and you have this way of thinking that the ligand will go to a receptor.
Yeah.
But do you try to find also intermediary support of transport, or do you think there is a possibility that maybe this secreted molecule could be transported together by two or in different complexes.
Yeah.
Or,
Yeah, no, absolutely. We don't know much about the transport system. And yeah, I mean, I think some of those proteins are going to be maybe some lipid type proteins can maybe, I mean, like the ApoLPs and so on, that might be involved in some of that. We have a project we just started now, which is to look at exosomes actually, because we believe that like the UPDs, those proteins do not go through the regular secretory pathway. And we believe that they are actually being loaded into exosomes. And so we are doing a lot of mass spec right now from purified exosomes to see what the content
in the blood,
yes. And we're trying to deal with contaminants right now because it's even so you enrich for the exosome, you still have a lot of blood protein that come down. So we can see proteins which are from the exosome, so we know they are there, but there are a lot of contaminants that we're trying to clean up. But that should help us to really get into this question about the trafficking, because especially at some point, some of those proteins have to go through. I mean, flies do have a blood-brain barrier, which are the glial cells, and they have to be able to access the brain in some cases. And we have no idea how they actually go through the brain. Yeah.
So, and we don't have an idea of the proportion of molecule in the blood that can be in an exosome or transported alone.
I think it's not.
By mass spec, you can maybe
well, that's what we're trying to do.
Difference between the protein in the blood that are just alone and.
I think if you Google fly exosome blood, I don't think you can find one paper yet.
Yeah.
So, but that's an important part about the trafficking.
And the choice to make it in the blood rather than to make it in whatever organ you could be interested in.
Yeah,
the rationale of it?
The rationale is that it's difficult to dissect the organs, right? And so we have, we've done it. So, like actually, the first paper we had on this proximity labeling, what we did was to express in fat, and then we basically cut off the legs of the fly because that's mostly muscle. But if you look in the thorax, you're going to get fat and muscle, so you can't do the dissections. So we can do it for a few organs where we can dissect. But what we are working on is another method actually of a double labeling. So we'll be able to, we have the secreted protein which are labeled, but then the idea is that once they reach a specific organ, then they are being modified again with a different modification. And we're using pupillation. It's another, so it's in this case you attach a specific peptide to the protein. So the idea is to label them when they leave and to label them in the organ, the destination. And this way you wouldn't need to do dissection. So that's one of the things.
Yeah, I have a question regarding the MEK inhibition stuff. Depending on when you treat your fly with the inhibitor, do you see differences in terms of phenotype reversion?
Yes, this. In fact, if you remember the slide, it was a 10 micron, whatever. It's only at a very specific concentration you see the suppression, because if you go higher, then you start to inhibit. There is some ERK signaling in the tumor. So then you start to affect the tumor, then you don't know what's direct. So.
But you didn't try to treat at different time points?
No, not this. It was mostly, okay, like that. It's telling us that we should be looking at ERK signaling. But, I also show that because you can do drug screens in flies. So actually, that's scary if you. I don't think people have actually spent enough time using the fly as a model for drug screening. Right.
Thank you.
Thank you. I don't need it.
On avait une trentaine de personnes en visio.
So
On va y arriver. Hello? So I think we are in English, correct? In English, okay. I just wanted to make sure that we are actually sharing the screen and that everyone can hear me.
Yes, your screen is shared.
Thank you. And you hear me well, I guess.
Yes.
Thank you. Okay. Well, I can see 3 people connected. So one, because I have to check those people that are present. I think there is 3 people. There are how many people? 8. Okay. Mariam, could you please share your, I don't know, your family name?
Yes, Motiola. So I will spell it MOTIOLAH.
Thank you.
Sure.
And you can see yourself in this list.
There is 2 people that are not supposed to be here.
That are not supposed to be here?
Yeah, Michelle, and they are not in our class. Yeah, and you, yeah.
I have no idea. This is the link that Bruno Segui
But they are not
gave
in
it
our
to me.
class.
They are not your class. But Madame Mariam, she's in your class. So I guess the link is the right one.
Yes.
I have no idea. Okay, I think we can start. Okay, so first thing first, I'm Giulia Leonardi. I'm a medical oncologist here in Toulouse. I work here next door at the AUCT. It's the Institut Universitaire du Cancer de Toulouse, Oncopole. I work in the clinical trials and immunotherapy unit, which means I see patients treated in the context of a clinical trial, all phases, mainly phase 1 and 2, but also phase 3, and people that are treated with immunotherapy either as a standard of care, since it's now a reality for many type of tumors, and also immunotherapy in the context of a clinical trial. So this is our agenda for today. And so I replace, of course, Professor Delors, who cannot be here today. So this is our agenda for today. So we are gonna go through stages of drug development quickly, and then we are gonna focus more on phase 1 trial, which are early phase trial. Some notions about biomarker discovery in this context, and let's bridge to phase 2 trials, which are not exactly considered early phase trial. Okay. Sorry, I have the keyboard, which is not working very well. Okay. So first, just to have a sense of what the drug development process is, this is a very long process. From the time we have a sense that a molecule can be useful, so we are really in the early discovery and development, so not even in the preclinical phase, but we are more in the pharmacological biochemistry, chemistry field of building up a molecule towards it, the time it takes to eventually being approved. FDA is the Food and Drug Administration.
Sorry to interrupt, but
Yes.
your screen stopped sharing.
Okay.
We do not have the presentation view. We just have the PowerPoint view.
Oh, I see. Let's try. So you have just okay, and so you had just the first slide.
Yes. Yes. Yes.
And now you see this way, because I can leave it like this.
I'm seeing the third slide right now, but still in PowerPoint view and not the presentation view.
So I can try to put on presentation, but can you see it well in the PowerPoint view? Because if it's not working, the presentation view, we can just leave it in the PowerPoint view if it's fine.
Yeah, right now it's clear for me, but I don't know if it is clear for everyone.
Okay, I'll share as a presentation and just interrupt me if it's not moving forward. Okay?
Okay, sure. Sure.
Thank you.
En fait, une fois que c'est lancé, arrêtez le partage. Aller sur Zoom. Faire partager. Et là, oui, voilà, c'est ça. Là, dessus, partager. Et normalement, en allant dessus,
we're gonna try. Just let me know. So I was just saying that this is like the time frame for a drug from a compound to be discovered towards being eventually approved for clinical use. It takes a lot of time. It means an average is about 10 years. It can take more time. It depends on how fast it moves along the process. There is an interest to, of course, shorten this period because we are talking about oncology. Sometimes these drugs represent the only treatment option for patients who are not responding to other drug classes that are already approved. So you can understand that for a promising drug taking 10 years, patients won't make it up until then. But so for drug development, we have a big, long part that is the discovery and the development of the molecule, all the preclinical research which is made on in vitro and in vivo research and experiments, and then there is all the clinical research part, which is made by 3 phases: phase 1, phase 2, and phase 3. Once this part of clinical research is completed, and several hundred, and I would say, yes, several hundred patients have been treated and results are positive. We will discuss what that means. The drug will be reviewed by the regulatory agency. This is just an American slide, which means FDA, the Food and Drug Administration, but it's the same thing for the EMA, which is the European Medical Agency, which review everything and gives his approval. What does it mean? It gives it approvals. It deems that the data coming from all this clinical research, basic and clinical research process are positive, so they have this drug have an impact on the life or quality of life of the patients. We are discussing, of course, about oncology drugs. And so the drug will be approved. In Europe, EMA approval doesn't mean that the drug will be used in each country, because in Europe, EMA gives his, let's say, his approval, but then each of the state, the financial covering of the drug is a responsibility of each country. And so, since we have a public healthcare system, each country has its own time frame to get the drug available for patients in terms of cost and coverage. And then there is the post-marketing, that is a safety monitoring, which means this is true also for other type of drugs, not only oncology drugs. Once the medication is in the market, there is still a monitoring about the safety profile, because of course very, very rare adverse events, so toxicity, are not necessarily visible while the drug is a clinical trial, because we don't have that amount of people that are treated, so in terms of probability. And so the post-marketing safety monitor will still check for signals of very rare toxicities. It's okay, they're sharing, I guess, now. This just to repeat a little bit, but so we have this non-clinical part in which the drug is tested in the preclinical setting, so in vitro and in vivo, and then it enters the clinical setting. In this clinical setting, we have a first part in which essentially we study the pharmacology of the drug, so all the pharmacokinetics and pharmacodynamics in human. We have a second part that is the therapeutic exploratory, which means we want to understand if this drug, once we have seen that is, we have understand this mechanism of action in human, or I would say we have confirmed what we have seen in the preclinical parts. We want to understand if it's effective. So if we can have a tumor shrinkage or at least a stabilization of the tumor. And then we have a therapeutic confirmatory, which means, does this drug do better than what is already approved or not? Because, I mean, if it's not better, we don't have any reason to use it. Across all this period, we of course are going to look at safety, and which is true across all these single parts, and efficacy is our goal only in more in the terminal part of the clinical research. As I was saying, the goal is to try to optimize this time frame because sometimes there are very promising drugs and people are in need. Of course, in oncology, this is a really, I would say, a true thing, which means patients sometimes don't have a lot of treatment options. So a promising drug that takes 10 years, as I already said, it's a little bit too much time. So the goal of clinical development nowadays is to try to optimize and so to make the passage across all these single steps of the clinical development easier in terms of not before phase 1, phase 2, and phase 3 were very separate type of clinical trial. Nowadays, sometimes we have phase 1 that can become phase 2 or have some expansion cohort. We will see to try to get more information on the drug, a promising drug that have some signal of efficacy, and of course it's safe from a toxicity perspective. What is the preclinical stage? I don't know what your background is. More biology here. Biology. So I guess you're stronger than me on that. Clinical stages, of course, all the part of in vitro and in vivo findings. It aims at understand what are the pharmacodynamic of these drugs. So what are all the on and off target effects? This is important because you can start to understand and have a sense of what the toxicities are of this drug, understanding what are they targets, and of course starting to understand what potential markers can be available and can be looked at to not only predict efficacy but eventually also toxicity. Why is important to choose the right preclinical model? Because of course you can get very different results if you get the wrong preclinical model. For example, cell lines, they give you kind of an.
I'm sorry to interrupt again, but your slides are not changing; they are freezed.
Okay, you are to the. Okay, I think I'm gonna put it in this. Yes, you were still where? At the 3.
Yeah, the 3 was freezed actually.
Oh, I see. Okay, I think I'm gonna leave it like that.
Yeah, probably better.
Yeah. So I think you guys can see it. Thank you.
Thank you so much.
Of course. So, for example, I was saying, so we are discussing about the preclinical stage. I mean, I don't think there was anything very crucial in the slides before. So about the choosing of preclinical model, why is this important? Because you can get different results or wrong results or misleading results when using the wrong preclinical model, of course. For example, cell lines, sorry, they give you from one side more of a stable, for example, genomic background or molecular background, because you have cell lines that should be homogeneous. But it's true, for example, that with cell culture passage, you can modify this aspect. So this needs to be kept in mind when you think about preclinical model to understand what is the development of the drug, and it's very important to make it, to align it with what your clinical question are in terms of the cancer you're gonna treat and the eventual toxicity that you want to focus on. Another interesting thing is that what is called reverse translational strategies. So sometimes you think that research goes only from the preclinical stage and then move forwards to the clinical stage. But nowadays, patients are more frequently biopsied, for example, at progression. So this helps clinician, but also researcher to better understand what is the biology behind, for example, resistance tumors in patients. So this is the example of EGFR TKI inhibitors. This is a drug that is used in EGFR mutant non-small cell lung cancer, which is a lung cancer that arises in a non-smoking people. So in the beginning, EGFR TKIs were used across all the non-small cell lung cancer patients without knowing their EGFR status. And so when looking at the results of the clinical trial, they see that there were a lot of non-responders, but there were some people that have incredibly amazing responses with tumor shrinking, a duration of response of almost 1 year, which is a big result at that time for this class of non-small cell lung cancer patients. So they wanted to understand what's different compared to the general population of lung cancer patients in these patients. And the reality is that these patients were carrying an EGFR mutation that make them sensible to this drug. So as you can see, it's not only discovering a drug and then moving forward, but also using data coming from clinics that can help the preclinical setting to better understand and to better shape the drug process. And this is again true for the second generation EGFR TKIs. So these patients, they understand that to respond to this drug, they need to have this mutation in EGFR, and these patients were responding greatly, but then at a certain point, this is about 12 months, they were progressing. And so these patients were having another biopsy of their tumor that was augmenting in size. And what they found is that there was a specific mutation in 50% of these patients that is a mutation that makes the drug not binding well. And it's a gatekeeper mutation in EGFR, and so a drug was developed to treat these patients that were EGFR mutated, but that were also carrying this T790M mutation, and this is osimertinib, which is a second generation. So this is just to highlight how the direction of clinical research is not only from basic to the clinic, but can be also in both directions. So let's go back to clinical stage of the drug development. So this is to give what are the most important point in each phase of drug development. So in phase 1, the most important thing is to find the maximum tolerated dose, understanding the safety of the drug, the toxicity, so the dose limiting toxicity. So this is the maximum of the dose that you can tolerate, and what are the toxicity that are the dose limiting. So what are the toxicity that are the most represented for this class of drug. In phase 1 trial, normally very small number of patients are enrolled because sometimes you have, we will see, what we call first in human, so drug that has never been tested. So you have some clue on toxicity, pharmacokinetics, and dynamics from the preclinical setting, but of course, you have to be very prudent before well, you don't want to enroll a lot of patients because you don't really know a lot about the toxicity. And normally, phase 1 trial, they don't have any registration value. What does it mean? That you cannot go to the European Medical Agency and with the phase 1 results and saying, oh, we have great results. So what do you think we can give this to people? No, because at this time, we can only say almost that your drug is not toxic, but you cannot statistically state that is efficient, that it has an efficacy on the tumor. So this is the goal of phase 2 trial. So the purpose is to define activity. Emphasis is activity. Endpoint is the response. ORR is for overall response rate. In this type of trial, the number of patients enrolled is augmented because you have already a sense of what the toxicity is. So, and you want to understand the efficacy. So, what we mean for response, overall response rate in clinic. So when you look at the tumor to say if your drug is working or not, you do normally a CT scan. So you look at the image and you have your parameters of your tumor at baseline. So before starting the treatment, you have, for example, a lesion that is 2 centimeter, and then you are gonna look, is this lesion stable? Yes or no? This is already good results. Is this tumor shrinking? Is this tumor reducing? This is a great news for us. Or is this tumor augmenting in volume? So is this tumor progressing? So what we are looking in phase 2 is at least a stabilization of the tumor, if not a reduction in volume. If the tumor continues growing, well, it's not defined as a good activity. And then, as I was saying, phase 3 trial, the purpose is to compare the drug that has been shown to be safe enough, that has a certain level of activity. When we compare it with SOC, which means standard of care, do we do better or not? And the endpoint are usually survival. So PFS is progression-free survival and OS is overall survival, which means does the patients have a longer survival with this new drug or not? Because at the end, this is what we are trying to do with these patients, improve survival and improve quality of life. And these are usually, phase 3 are usually the trial that are used for registration. So again, phase trial has these 3 main objectives. So assessing safety, is this drug safe enough in human? So what are the adverse effects? What are the toxicity? What are the terms of tolerability of this treatment? Another important element of phase 1 trial is to determine the dosage. So in phase 1 trial, as we will see, the goal is to identify what is the maximum tolerated dose, to understand what are the toxicities associated with this drug, and to define the recommended phase 2 dose. So what is the dose that will be used in the phase 2 and then eventually in the phase 3 trial? And then, of course, understand all the pharmacokinetics, so how the drug is absorbed, distributed, metabolized, and excreted in the body, and also understand if the drug needs to be taken with food, not with food, if there are any major interaction with other molecules. So I mentioned this before, there are some drugs that have never been used in human. These are called FEH, first in human, phase 1 trial. In this trial, you need to find the dose with which you want to start to treat your patients. In this context, you take into account different elements. One thing that needs to be always considered, at least in oncological clinical trial, since these are patients with serious disease, you want to try to give a drug that has at least a minimum of efficacy. You don't want to give a placebo in the context, something that it has no, that is not gonna work. Of course, this needs to be made in weighted with the toxicity, but you want to try to start with a dose that have at least in the preclinical setting sense of minimum of activity. Of course, the choice of this dose depends on what are the data that have been obtained in the preclinical setting, and so also all those that are the predicted safety risk in terms of of target and on target effect, if the drug has ever been used in other contexts, and if it's an immunostimulator or another type of drug. What does that mean? That there are some drugs, for example, TKI, that when you stop them, then you have your washout, and then, well, the drug is not there anymore. But there are some nowadays, like immunomodulatory immunotherapy strategies that activate a cascade of events. And so it's not that when you remove the drug, which usually have a half-life that is much longer. If we discuss about monoclonal antibodies, we are about 3 weeks. So at that point, you need to be more cautious, because you don't have that space of interrupting the drug and slowly making the target not targeted anymore. When we look at first in human, also we need to think not only to the toxicity. What is a toxicity? Toxicity is, you have your liver enzyme that increase, or your renal function is not doing great, or you are gonna get anemia or stuff like this. But you also need to understand, for example, if you have any teratogen effects, if you have any toxicity at the reproductive level, if sometimes you can also have some carcinogenity type of risk. For example, there are some chemotherapy or PARP inhibitor that have a risk to that may increase your risk to have a second type of cancer. So usually when you look at this phase 1 trial, you have a small cohort in which you do dose escalation. So you start from this initial small dose that you have considered and decided depending on all the factors that I come to mention. And then from this small dose, you start escalating, so augmenting the dose until this dose is MTD, so the maximum tolerated dose, or if there are some dose-limiting toxicity. And this is done to define what is called the MP2D, which is the recommended phase 2 dose. That is the dose that will be used for phase 2 and phase 3 trial. In the phase 1, you can also have, and this is starting to become more frequent because of the reason that I mentioned in the beginning. So with the attempt of optimizing the development of the drug and be more efficient. So within the phase 1, you can have expansion cohorts. When you find your MP2D, you can start to treat more patients to try to have more of a sense of the global toxicity and have some preliminary anti-tumor activity. So we are a lot saying what is a toxicity, that we are discussing about toxicity. What is toxicity? So toxicity are adverse events. So any events during treatment that can have an impact on the patients or not, but that can be related to the drug. So this is just a snapshot of what is the table for CTCA. This is the common terminology criteria for adverse events, which is using all the clinical trial. So for example, you use a drug and you can develop anemia. Your anemia can be a grade 1, 2, 3, 4. You want that to be a grade 5, which is death of the patients because of anemia. But you can have a several degree of this toxicity, and this is true for a lot of elements that are not only biological laboratory, but you can have, I don't know, you can have gastritis, you can have anything. This is a very exhaustive list, and each element is graded. Normally, grade 1, 2 are mild event. Grade 3 starts to be a serious event. Those limiting toxicity are toxicity that start to be grade 3, 4. And usually you don't treat when you have toxicity at this level. With grade 2, it depends on the type of drug, and normally with grade 1 toxicity, you can keep treating your patients. So we were saying we start from a dose and then we do dose escalation. But how we do dose escalation? There are a lot of let's say model to do that. The 3+3 design is the most let's say the traditional design of a phase 1 trial. Essentially is the traditional design, and also the more conservative in terms that really gives you the time to understand and to observe the toxicity. So usually first in human have this base of design. We will see nowadays this is integrated with EA or other statistical methods that can integrate more elements to build this trial design. But 3+3 is really the basis of phase 1 trial. So these represent almost the same thing. I mean, the same thing is just represented differently. It works is like this. So you have, for example, your first dose with which you want to start, and you enter only 3 patients in that dose. Okay, you start to treat them. And you observe them for at least 2 weeks. And then if you don't have any toxic response, so you don't have any toxicity, you can escalate the dose, okay? And then you enroll other 3 patients. And then you wait another 2 weeks to look at the toxicity at that new dose. And then if you have a toxicity at that new dose, you enroll 3 patients more at the same dose, okay, to understand the frequency of that toxicity. If everything is good, then you can move forward until you have essentially less than 1/3 of toxicity for that dose level, okay? So I wrote it down here. So the DLTs are usually relevant toxicity that are grade 3 or higher in the CTCA. And when you have at least 2, then usually you don't move forward in the dose escalation. And then you define this is the maximum tolerated dose. So when you have 66% of toxicity in your cohort, and then you pick as your LP2D, so recommended phase 2 dose, the dose level before. If something is not clear, you just
we already saw that in the first class. Right?
Oh, I see. So, so this is the Fibonacci method, which is a way to define the dose. This is when you already have predetermined dose escalation. And with the Fibonacci methods, essentially your escalation the amount of drug reduced once you move forward in the escalation. So in the first level, you do a dose escalation of 100%, and then you reduce moving forward in the dose level. And then you can have also an adapted and adjusted escalation, which is associated with toxicity. So if you don't have any toxicity, you move forward to 100%, so you double the dose. If you have low-grade toxicity, you can do a 50% increase of the dose. And then if you have higher toxicity, you just stop, or you can do a dose escalation 25% more. I won't enter in this because I will just mention that they exist because I am not a statistician. But this is just to say that 3+3 methods is, you know, like really the standard, the base of phase 1 trial. But nowadays there are a lot of model-based approach and based on the risk assessment, and so you define the dose label integrating a lot of elements, including pharmacokinetics, pharmacodynamics, and the toxicity element. Dose expansion is a moment in which you have found your LP2D, and then you want to understand if really the toxicity profile is what you have observed. Those expansion are also useful if you want to improve your understanding about pharmacokinetics. For example, if you need to take it when you have, you know, eaten or not, or if there are some association that you need to avoid, because this gives you the opportunity to test that dose that you want to move forward in the clinical development into a higher number of patients. So in the end, when you need to choose your dose for the phase 2 trial, you need to take into consideration different elements, which are, of course, the drug class, the mechanism of action, what is your patient's population. Of course, if you have a biomarker selection, this can help you, because if you have a biomarker that helps you, I mean, we did the example of the EGFR, a drug that is potentially very helpful. If you test it in the wrong population, you just are gonna miss its efficacy. The toxicity of the drug, so dose limiting toxicity. Once you observe the drug in the population, are there any chronic toxicity that require, for example, interruption, pharmacokinetics, pharmacodynamic, and of course, trying to move forward those that have a clinical activity. So this is an example when you use a pharmacodynamic biomarker. For example, you know that your drug is gonna target a specific mutation, but for example, you know, you have a molecule that has no interaction with the target. Well, this drug is not gonna move forward, so you're not gonna test forward this drug. But then you have, for example, a drug that you know pharmacodynamically that has an interaction with his target, but you don't have any downstream effect, which means you don't see any modification, for example, in its pathway. And so you have a no-go, so you don't move forward in the development of this drug. And then you have a drug that has an interaction with his target, has a downstream effect, but has no anti-tumor effect. So you don't see any shrinkage, what we were saying, or any control. What do you do with that drug? These are the challenging. I mean, the last line is just the better scenario in which you have a drug that interacts with its target, that has downstream effect, that shrink the tumor. So you continue to develop this drug. But when you don't have any tumor effect, are you really looking at the right population? Are you missing any biological effect? So basing your decision only on pharmacodynamic element can be difficult and sometimes can be misleading. So when you move forward in the phase 2, you need to take into account the globality of elements. In the phase 1 trial, sometimes we have these two type of clinical trial, which are the umbrella trial or the basket trial. Most commonly, I would say phase 1 trial are basket trials, which means that you treat in the same trial different type of cancer that have a common, for example, genomic alteration. Or that you have seen in preclinical data that, for example, you have data that supports the use of this drug in, for example, colon cancer, pancreatic cancer, and bladder cancer. Okay, and so in phase 1, you have sometimes, I would say, it's pretty common to have this type of structure. So you have different cohorts, but you treat the patients with the same drug. And then the umbrella trial are a little bit on the opposite, but almost the same concept. In this case, you have one type of cancer that may have different type of anomalies. For example, let's take lung cancer. You can have EGFR mutation, you can have ALK rearrangement, you can have ROS1 rearrangement or MET exon 14 skipping mutation. So you have different cohorts, and then you treat each with a different drug that is based on their specific genetic context. Biomarker discovery is a very hot topic in oncology development and in early phase trial development. This because biomarker helps you understand not only the tumor, but how the drug interact with it, and also gives you elements to predict which are the disease or the context in which the drug will be more efficient, and also can help understand toxicity or predict toxicity. And nowadays, in phase 1 trial, this is a very important element of the phase 1 trial itself, because it helps understand how the drugs works and how it works against the tumor. So, for example, these are all the biomarkers that were used in this phase 1 trial with an AKT inhibitor. So they were using circulating free DNA to study resistance. So they were looking at circulating free DNA to understand, while on drug treatment, if they were able to find circulating free DNA before seeing the tumor augmenting volume. Okay. You can use all these biomarker for toxicity monitoring, so looking at the pharmacokinetics parameters and try to understand which is the best drug level to expand. You can have genetic and immunohistochemistry studies to understand better this drug development. So sometimes these elements, they look perfect. So these are this type of graph are called waterfall plot. So each of these bar is a patient, and when the when the bar goes down, it means the tumor shrinks, and when the bar goes up, it means that the tumors grow. So these are genomic abnormalities in tumors that are predictive of response to a drug. On the left, we have BRAF mutation in melanoma that lead to the approval of vemurafenib for the treatment of this disease. And on the right is crizotinib for ALK rearranged non-small cell lung cancer. So you can see that it's the same discussion of EGFR. Patients that were carrying these anomalies, they were amazingly responding to the drug. Okay, so it's very nice when you have, you can really identify your population and not just, trying in. We all know nowadays that, tumors is a very complex biology. So when you are able to target a specific abnormalities, this is great. And then sometimes biomarkers as BRAF mutation or ALK arrangement can be also used as a prognostic biomarker. They can be predictive. You know the difference between prognostic and predictive. So prognostic is a biomarker that gives you a sense of the aggressivity of the natural history of the disease. And predictive is a biomarker that gives you a sense of how if the patients will be responding to a drug having that type of alteration. Sometimes this is not clear. For example, BRAF can be prognostic. We have element of pharmacodynamic activity. It is predictive. Yes, it depends on the disease, which means melanoma, they respond to BRAF inhibitor when they have a BRAF mutation, but this is not true, for example, for colon cancer. Okay, so it depends really on the context they're used. This can be the disease, or it can be it's essentially, yes, the type of tumor. So we have a biomarker for response, but there are also biomarkers that can be available to understand those patients that won't respond to a specific treatment. For example, we know that KRAS mutations gives resistance to EGFR therapy in colorectal cancer. So this is just to highlight how not only we need to understand who are the patients that are responding, but potentially also those who are not responding to cancer drug. And then sometimes we have biomarker that they looks perfect, but because we can see when we give the drug, we have some nice Western blot, some reducing of the phosphorylation. We have this TKI, very nice, reducing the activation of their target. But at the end, when we use them in the patients, well, they are very toxic, and we don't know that until we test them. And so this is the case of this tivantinib, which is a MET inhibitor that has a very nice activity on its target, but it's toxic on the tubulin. So it works almost like vincristine, and so patients do not tolerate it. We're almost at the end. A big element is biomarker in the context of immunotherapy and the tumor microenvironment. We do not have yet a good biomarker for giving immunotherapy, and immunotherapy is really exploding, and immunology is a very complex and dynamic process. So we really need to understand what are the mechanisms that essentially drive immune resistance. Some patients do respond and then they will develop resistance, and we don't know why. There are some patients that are resistant in the beginning, and so there are different elements that can be taken into account. Some elements are in the tumor microenvironment, some elements can be in the antigen presentation machinery, other elements can be genomic elements. So this is how it can look, an immune monitoring study. So patients that comes, for example, for receiving treatment or because they have received treatment and we want to understand what is the evolution of their immune response. So for example, we can do immune monitoring by looking at PBMCs, so peripheral blood mononuclear cells taken into blood samples, or we can look at it in the tumor, for example, looking at tumor infiltrating lymphocytes or other cellular components that are infiltrating the tumor, if we can get a tumor samples. And we can do this actually in any liquid. We can do ascites, we can do saliva, we can do any biological compartment. For sure, blood is the easiest one compared to to tumors, since tumors you need to biopsy it, and sometimes this is, I mean, you cannot do it every week. It's not ethically acceptable. So for sure, you may want to have a samples in the beginning to have a baseline. You may want to have one intermediate, and then having, for example, a tumor sample at resistance, at progression, to understand what has changed in the context of the tumor microenvironment compared to your baseline treatment. And then you can have more easily blood samples to look at the evolution over time in the periphery of your immune system. So yes, for all the reasons that we have discussed in here, and actually biomarker represent a big part of phase 1 trial. And nowadays, I would say patients treated in phase 1 trial, for example, in the beginning of the treatment may take like 1 hour for infusion, but they have like sometimes 4 hours of observation, even 24 hours. They are hospitalized because you want to do all the PK, blood drawn, you want to do all these immune monitoring process. So this is done from one side for their safety, because for immunotherapy, mostly sometimes you can have some cytokine release syndrome, so you want to monitor them closely. But the other part is also trying to have all this information about pharmacokinetics, dynamics, and in terms of immune monitoring. And once all this part of the phase 1 trial is done, well, phase 2 can be started. Phase 2 are built statistically to understand the efficacy of the drug. So does this drug shrink the tumor? Does the tumor stay stable? And also, while on phase 2, of course, you continue looking for the safety profile. So to monitor toxicity, you start to enroll more patients. And so you can start to see some more rare toxicity. So this you need still to be aware, and you can also better understand what is the context of dose ranging and how to administer it. So to refine, let's say, what you have been observing in phase 1, because then phase 3 trial will be the moment in which your dose will be the one that you are gonna test against your standard of care. So you want it to be safe, to make a good comparison in terms of toxicity, and to be the right dose to have your biological effect to see your tumor shrinkage and so perform better than the standard of care. And I think we are done. Yeah. And so you had already this course.
Not really all the things, but
Some of them.
Yeah.
Mhm. And because you are a master too?
Yeah.
Yeah. Okay. And so the other course was.
It was clinical trials course, but
Okay. So there was a little bit of phase 1. Of course.
pharmacokinetic and
Okay. A little bit of
and
overlapping.
also pharmacoepidemiology.
Okay. Cool. Okay. Any question here? Yes. Can I stop it? There are no questions. Thank you. Bye. Bye-bye.